Okay, then let's continue. Or you can leave if you don't mind, you can leave some windows open in the back. If you don't mind. I think it's quite warm, but anyway. So, visual evoked potentials maybe so far have sounded a little bit boring from the cognitive perspective, but of course a big field or big paradigm in terms of the research paradigm in event-related potential research is of course to see what kind of factors modulate these event-related and primary sensory event-related evoked potentials. And there are different forms of modulation that apply, and here's just a small selection. Of course, I would argue that the literature is, I guess, quite biased in this regard. So, because if you find that your favorite factor modulates visual evoked potential for the longest time over the last, I don't know, 40 years or so, you will have published that. And if you found that something does not modulate the evoked potential, then this will have ended up in a file drawer. So that's more a problem of the academic system than anything else. But so we definitely know what kind of factors modulate evoked potentials. We have wasted essentially all of the knowledge in terms of factors that don't modulate event-related potentials. It's kind of a little open science side note. So let's look at what has been found. And what is kind of a general factor that affects essentially everything is arousal, whatever that should mean. And what you can see is that with higher arousal, you observe stronger responses. So the manipulation here was medium arousal, respond as quickly as possible. And the high arousal was, I guess, based on some social reinforcement, basically telling the participant you didn't really respond that fast currently, respond even faster now. Do the study questions. You hate me so much right now. There you see strong responses. This is actually a common finding. Also in fMRI, I would say you find the same thing. And this is also why in general you want participants who are alert and actually interested, and you don't want undergrad students who come to you on a Sunday and at a party yesterday, and then they go to the scan or something, and you know it will not turn out well. There was a question.
Yeah, but it's not really the stronger response, it's a more positive one, right? Because
you mean this one?
Less strong.
No, that's true. Yeah, you could also say there's something funky with the baseline, or you could say we are actually only interested in the P1 response, and then we find a stronger response. But yeah, you're right. There are then different ways to quantify the difference here. One thing is also to look into the area under the curve across the whole thing. And here then I would argue again that here, although this is a negative contribution going from the dashed line to the thick line, but because this is more of a difference here, that it's in this area under the curve, it's actually a stronger response. Yeah, but so this is basically just to remind us that we want alert and participants so that we record some sensible results. There are other things that evoke visually evoked potentials. One thing is spatial attention. This is from our own work, where the P100, you can ignore this, that was a perception decision paradigm, where the P100 is modulated by spatial attention. So people either knew where the stimulus was coming up and attended to that side of space, then you get a stronger response. As compared, they didn't know where the stimulus was coming up, then you get a weaker response. There's actually the more standard paradigm is to contrast attended versus inhibited stimuli, then the differences are even stronger. So essentially this means if a stimulus is showing up in a location of the visual space that people are attending to spatially, then you observe a stronger response. A stronger response than if people are not attending that point in space. That also generalizes to fMRI. This also generalizes to, of course, invasive electrophysiology. So attention and usually in terms of the signals that you observe, it what's the word that I'm missing? It does not emphasize. It increases. That's also not the word I'm searching for. But anyway, it enhances. I think that was the word I was looking for. I think it enhances the responses that you can observe. And here's another example which actually interrelates adaptation and attention. Let's see whether I get this correct. So in this thing, there was an adaptation condition. So a stimulus is preceded by a stimulus at the same location. And then there's a no adaptation condition where a stimulus is preceded by a stimulus at a different location. And then this can either happen, so this, I guess it was a 2x2 design where then attention was also allocated to that location in space or not. And what do we observe? For attended preceded by unattended, we see a P1. For the attended preceded by attended, the P1 actually comes out to be roughly the same, but we see a difference in one component. So if an attended is preceded by an attended, we see more of a adaptation of the N100 in the sense that it's a weaker response than if something is attended but was preceded by something that was unattended. So you can essentially, what this is an example for without being completely clear about the logic of the experiment, is that adaptation effects are there. We've already seen that in the checkerboard responses that I've shown you, where the response to the stimulus coming up was much stronger than to the reversal. And then you, of course, can cross these effects. Another cognitive modulation, if you want, is the N170 or vertex positive potential. It's a cognitive modulation in the sense that you do use a specific stimulus material. So it's not really cognitive in the sense that it's an internal process like attention, which of course also is kind of externally cued, but here it's really in the stimulus and mapping essentially the findings, mapping to the findings in fMRI, where you of course know that there's something called the fusiform phase area, so a specific part of visual cortex which is strongly responsive to phases. There is in EEG research, there's the so-called N170. It's called vertex positive potential also because people mainly, most people look at it when they discovered and studied that at the vertex, which is this location at the highest point of your skull. So if you look at that here, you see something positive happening at 170. I always view it like kind of a delayed N140 because essentially you see a peak and then you see a negative peak, which is a little bit delayed with respect to the standard N140. But then I mean, on the one hand, 30 milliseconds are quite a lot in EEG research. Then on the other hand, if you change the stimulus material, the peaks, they also shift around a little bit. So so I wouldn't say that N170 is something completely different from N140, but it's definitely the idea is that it's the response that you observe for phases, in the sense that you observe a stronger response here at 170, or a larger response here at the vertex for phases versus cars. So here you have phases versus cars. And then if you look more into the lateral regions, then this is, you see the same thing as negative. And again, you have the N170, its negative name at 100 milliseconds. So this goes again into this direction, that you have the same thing being projected to the vertex, and it's called a positivity, or being projected to the lateral occipital electrodes, then it's negative. So so phases are special, which of course from an evolutionary perspective makes a lot of sense, and this also has of course given rise to a lot of work on phase perception, and you can basically EEG and MEG also give you kind of a marker of phase responses, as fMRI does. So if you always thought fMRI would be a good idea to do fMRI, because then at least you get something phase specific. You can also do that with EEG. I don't think there's that much literature actually on trying to find that the fusiform phase area only is actually giving rise to this vertex positive potential. So that would be a question of EEG-fMRI integration. I don't think that exists much, because all the work on phase perception in cognitive neuroscience is mainly done in fMRI. And this having said that, of course there is work on phase perception using EEG, but it's definitely a smaller field. At least that's my perception. Right. Good. So this is then the summary, and I think for the other, this is why I was confused in the beginning, I think for the other event-related potentials, the summary comes up first. Here it comes up last, which, because it's a summary, makes also sense. So one thing to learn about these event-related potentials is definitely the component names, what they relate to, so what the ideas, where they come from, and then maybe a little bit how one can modulate them. So we've seen that the C1/ or aka N70 is largest in posterior midline electrodes. Well, I haven't explicitly said that, but we looked at this one electrode, and it makes sense because it's coming from primary visual cortex, which is in the midline. It's probably generated in primary visual cortex. You will always find with all the event-related potential that I say probably or most likely or something, because in the end, something that you would have to do is actually to have, essentially you would have to have electrodes at all places inside the brain, and check that all the electrodes are silent except for the electrodes that you have in a specific area, and then look at the EEG. At least you would want to have an electrode in primary visual cortex of the participant and the EEG electrode, but of course you will not have that. And I think although you have electrodes in patients, sometimes in EEG electrodes, I think I've never heard that for medical reasons it was necessary to put an electrode into primary visual cortex. Might have happened, but I think I never heard of it. So this is why I always say it's probably generated there, because you can only infer that from source reconstruction essentially. But don't forget that fMRI images are also reconstructed images from K-space. So fMRI is not better. So the polarity varies for the C1 and /N70 depending on whether you're in the upper or lower visual field, and there was this explanation with the dipole. For stimuli on the horizontal midline or everything that, or full field or full hemisphere presentation, you cannot see it anymore. And it's actually, what then I also keep here, so the onset latencies, and you see some variability, and the peak latency, you also see some variability. But this is really the first response. The P100, which is for most of the stimuli that you look at, that I look at, which are checkerboards, the really first response that you actually see, that has a little bit of a later onset, is stronger over occipital electrodes, and as we've seen, is generated in dorsal extrastriate cortex, maybe a little bit of ventral fusiform gyrus, so going to the temporal lobe, and is modulated by things like sinus conscious spatial frequency, arousal, spatial attention. And the N140 is, one can maybe, so this is what I put in here, which is also based on the Russo paper, maybe one can dissociate subcomponents of this peak, but yeah, maybe also not. And it's negative over these occipital lateral electrodes. The generation is not that clear anymore for these things, so you cannot, like for C1 and 70, say, okay, this is likely B1, but it's more kind of everything in visual cortex, maybe a little bit of parietal cortex. And then finally we looked at the N170, which is phase selective, and you either look at it here at the vertex, or you also look at it the electrode sites. So these are, of course, these summary slides, they are really to help you to memorize things. So not that easy, nice to look at, but if you have these things kind of in your head, then you will know what people mean when they say, but I guess you get a P100 there if you use the stimulus. Then you know what they mean. Good. Questions about visually evoked potentials at this point? No. Good. Then, yeah.
I think it's a very general question, but when you showed us the pictures with the electrodes, the big one, when you showed the checkerboard on the left side, in the very beginning, with the 64 electrodes, are we similar? I'm sure you are, but are we similar to how we extrapolate from the electrodes to the area around it?
You mean this one?
Yeah.
We're not going to talk about the computational stuff that you use for that.
I can very generally conceptually say how that works.
Yeah, sure. I mean, the easiest way is, so for example, if you focus on, let's say, this electrode here, then you can, there are different ways, but if you want to spatially interpolate that, then you can, of course, ask yourself, what are the electrodes around it? So what are the electrodes around? Then you can, for example, define like all the electrodes that have the shortest distance in all directions of space. So, for example, this electrode, this electrode, this electrode, and this electrode. And then you have, of course, observed values here, and you have observed a value here. So you have like 5 values, and you're now trying to explain how what you have in between. And a very simple thing would be then to do a weighted average that is a function of distance. So you use a weight that, so you do a convex combination. So with my where's my pen? Oh, I have a pen here. I have a digital pen. But where's my pen? Anyway, hopefully. Oh, sorry. What's it doing there? Does that work? Yeah. So then you can do, if you have your one electrode, YC, I don't know, electrode 1 plus 1 minus, so you can do a convex combination where W is in this, and then you make this weight a function of the distance, so you have it set to 1 if you are here, and then you let the other one come in, so that it's basically corresponding to this. And then you at least can do some interpolation here. That would be a linear interpolation, but of course you can do much more fancy things. And then you of course also have to keep in mind that you not only do this for this one electrode, but for all the other one and all the other ones as well. And then it's the question how you combine them. Whether you do something like this and then add them all up over space, or do something else. So this is kind of a spatial interpolation for that. Actually, I never really went into the details of that, because I use these pictures primarily to look at and not to make any scientific claims. So it's just, you could also not do that. I mean, you can also in your head try to, I mean, you could also just plot the electrodes very, just make the electrodes bigger and make them red, and then you would have a bunch of red electrodes here and a bunch of blue electrodes here. So that's the story about that. But you can look into the details, of course, how this is interpolated if you look into field trip and so on, how they exactly do it. Something that doesn't fall into probabilistic models, so something that I don't get into that much. Any other questions? No. Good. So then the next next sensory event-related potential are somatosensory evoked potentials. So if you work with me or Radek, you might record visually evoked potentials. If you work with Felix, you might record somatosensory evoked potentials, because he has done that a lot and he still does. And I also did that with him. So somatosensory evoked potentials are, of course, potentials that you record to some form of tactile stimulation. And this is what the setup that we used for this Bayesian surprise paper with Felix from my first postdoc, I should say. Although I always think that I only ever did one postdoc, but formally I did a second postdoc. Anyway, you're not interested in that. This is a typical setup for recording somatosensory evoked potentials also in the clinics, and they might be even more used in a clinical setting than the visual evoked potentials, because they are also used to monitor anesthesia depth. The way it works is as follows. You have a little electrical stimulator here, in this case 2 electrodes, and then you can press a button and zap the participants. It's not the most comfortable feeling, but also you don't die of that. So yeah, and what you're trying to stimulate actually with this setup is this median nerve, which is the nerve that innervates your fingers, essentially. So it's a big nerve that is hidden under your tendons at your wrist, and you try to stimulate that. It feels, this electrical stimulation, a little bit like when you touch an electrical fence, but much weaker, because these electrical fences, of course, they are there to keep the deer wandering off, so it should hurt a little bit, like the study questions. And here you don't want to hurt really the participants, and you don't want to really induce some pain. So that's the setup, and of course this electrical stimulator can be driven by MATLAB or anything else, and also whenever it gives provides a stimulus, you can record that and get a timestamp. And then what you get on the electrodes, and at this time I'm fairly certain that this is from 64 electrodes, you get here overlaid a response where it shows some peaks that are of interest to somatosensory event-related potential researchers: the N20, the N45, the P60, and the N120. You also see already a pattern if you compare that to the visual responses. The later responses often are actually a little bit more, a little bit stronger, and then the problem is always with the later responses. Although they are strong, you don't really know where they're coming from. The early responses are a little bit more confined in space and time, and you know where they're coming from, but they're also weaker. We will talk about this. This is another way to represent what you got from a grand average. I think it's better to show something like that than to show a single electrode, because first of all, you record it from a lot of electrodes, and it's not that you step one electrode there and then all your data collapses to this one electrode. On the other hand, of course, it's not that clear to see what's actually going on. And here the stimulation is really short, so that's 0.2 milliseconds, so that's really, really short, and was with 5 microampere. Of course, you can increase that, and then you get stronger responses, or you can decrease it to do consciousness research, and then you are hard-pressed to see anything. This is a reminder of the functional anatomy of the somatosensory system, which you may or may have not studied. So let's see how well versed I'm still in that. So definitely the first neuron in the afferent pathway to the brain sits in the dorsal in the ganglion cell, which project to the dorsal root. So you have a nerve cell that's actually sitting in the bones next to your spinal cord, not in the bones, but in a little bit of a cavity. And then the second neuron sits a little bit higher up, so at this point at the medulla. So this is where some relay takes place, and then a third relay or a third neuron sits in the thalamus. As you know, the thalamus is the gateway to the cortex. Then it's relayed, and then it is projected to a primary somatosensory cortex. And of course, the EEG responses that we are interested in, we record from somatosensory cortex. But you should not forget that in contrast to vision, the tactile responses that you record, they can be from closer to the brain or from further away from the brain. So for example, if you stimulate the medial nerve here, it's closer to the brain. So you might think that you record something earlier than if you record it here, especially if you record from the feet. Of course, the way to the brain is a lot longer, so you might observe a little bit of a delay of your first somatosensory response in the brain. Now, if one looks into the, you always have never seen this, but anyway, if you look into the brain, you see the posterior bank of the central sulcus. So you have the central sulcus there where it points, and I cannot point here. And then in the posterior bank you have the primary somatosensory cortex, which of course also has some subregions. And for example, Simon Eickhoff has done a lot of work on trying to establish these areas also in the human brain based on fMRI. And one thing that is, or takes some getting used to, especially if you come from vision, where secondary visual cortex is right next to primary visual cortex. Secondary somatosensory cortex is actually more down here. So primary somatosensory cortex extends all the way here and corresponds to what you, of course, have seen as this homunculus. Maybe there's a picture of that later. And secondary somatosensory cortex is actually this region down here. I was quite confused about this for some time because I always thought that, okay, here's primary somatosensory cortex, then like in vision, the secondary should be right next to it, but that's not the case. Primary somatosensory cortex then also can be actually further decomposed into area 1, 2, area 3A and 3B, where I think these are Brodmann areas again. With slightly different functionalities. So, but the main thing I want to remind you of is that primary somatosensory cortex extends along the central sulcus, and yeah, if you do fMRI of this, you can actually see that you can recover this homunculus if you. So you can dissociate foot and hand areas. At least that's what the people from the Blankenburg lab say and show. And I believe them, of course. In EEG, we have, I think they also recorded EEG data by now, definitely from hands and feet and so on. In this case, I focused on median nerve. And so I only have data from the hand. Essentially, or medium nerve, so what do you see then? And I've already showed you the overall response. Now, basically, in analogy to what we've seen in vision, going over the responses in somatosensory cortex, and here, the earliest cortical potential that you can see elicited by median nerve stimulation is called the N20, so it's a negative response over the electrodes over somatosensory cortex. I will show you the topology in a second. And the idea is that this is coming from S1. So it's a little bit different than in the visual system, where if you give a very simple stimulus, like the checkerboard, you only see the second response. Here you give a very simple stimulus, namely an electrical pulse to the median nerve, and then you actually see this S1 or primary somatosensory cortex response. Which more specifically is actually area 3B in the posterior bank of the central sulcus. And it has high relevance for clinical diagnostics. Also, if, for example, in neurology, if you have these syndromes where, for example, the Schwann cells disappear in your peripheral nerves, then you get these connectivity delays, and so you can use this as an early sign for onsets of this. So this is really something that is used clinically, not only in our happy cognitive neuroscience world. Then, so that's the first peak, and Felix will make sure that if you record somatosensory potentials that you can see that. This is also with these early components. They are good quality assessment tools. So if you set out, so you want to record somatosensory evoked potentials, but you don't really know how to position the electrodes, and it's your first time recording EEG, or it's a new EEG system, then this is actually something where you should be able, also for yourself, be able to reproduce things like N20 or P100 before you do anything else. Because if you don't see an N20 or not see a P100 for a checkerboard stimulus / a median nerve stimulation stimulus, then in this case you're actually doing something wrong. I'm not a person who would think that if you don't see a response to a subliminal stimulus that is presented under a non-attended condition in Chinese calligraphics compared to Norwegian calligraphics, that you really should see something there, or you will get not your master's degree, but you should see these kind of early responses. Now the P145 and N60, which are a little bit later, they you see them contralateral and frontal electrodes, and that's not really clear where they are coming from. And the N120 central and temporal ipsilateral contralateral electrodes, this is most likely from secondary somatosensory area, so from these things that are more towards the Sylvian fissure or towards the temporal lobe, but also this region is somewhat debated. But if one looks at the responses, one actually believes that a little bit more. So this is again the data matrix view, where you can already see that a lot of stuff is going on, and you also see again that for some electrodes you see something positive, for others you see something negative, and you have this first peak. And actually here the N20 is actually, let's zoom in there, the N20 are these weak things here. This is N20 and this is already P60, so the first response is relatively weak here. So let me show you that in the more intuitive way. So we are now again going through time and look at all the electrodes from front to back and right and left. And this was left median nerve stimulation. So again, we used this laterality of the whole system to also see some laterality in our responses. Now we switch forward to 20 milliseconds. And there you can see the fairly okayish dipolar pattern. Here's some positivity, here's some negativity, and here over these electrodes, this is then most likely where the dipole actually sits. So you see that it sits contralateral, and then it sits also much more frontal, of course, than we've seen for the visual response. So it sits essentially where you would imagine it to sit. So after some fiddling with the great biosemi system that the Blankenburg group uses, I'm convinced myself that I can record an N20. But it's not the strongest response. Actually, if you look a little bit later at N140 P60, this gets much stronger. And if you then go to later times from the N20 and then give even longer towards something around 140 seconds, you see that both sides of the brain become active. Yeah, so this then leads to the idea that the somatosensory stimulus is first processed in the contralateral hemisphere, and then once it's processed, whatever that means, in the primary somatosensory cortex, it's sent for further processing. I don't know what these terms mean, but people talk like that. For further processing, it's sent to both right and left secondary somatosensory cortex to figure out what the stimulus was. Good. I will not be able to finish auditory, or oh no, I do it. I do it quickly because the story remains the same. So we can actually in the next 5 minutes finish auditory evoked potentials because the story is very similar. So this is data from the SPM website. So you find an auditory data set on there, and you can use that to familiarize yourself with EEG data analysis in SPM. This refers to a bi-oral representation, so they had tones sent to both ears and then recorded from the scalp. So here you wouldn't assume some lateralization effects in the EEG because you stimulated both ears actually, and this is the typical setup for auditory auditory evoked potentials. And then again, you see like something as P1 and N1 for the interesting electrodes and later responses. here, one thing that is important, and that I now have to squeeze a little bit in, is that you can record auditory responses from the brainstem. And these are actually used in infant diagnostics to check whether they can hear, because you cannot ask an infant, can you hear, do you understand what I'm saying? But you can check whether this actually, whether at least the afferent pathway works by looking at the brainstem potentials. You will find more about that in the luck book. We are not interested in these brainstem potentials. We are interested here, at least in the response we get from cortex. And there's, as we just discussed, the auditory brainstem responses. Then you can get something a little bit more from subcortical stuff, and then what we are really interested in are the cortical responses, which gives you a P1 around 500 milliseconds, frontocentral electrodes, and N1 with possible multiple subcomponents. As you see, I'm rushing a little bit because we have to get out. And this shows you the data matrix. And this shows here the P1, now not lateralized because the stimulation worked on both sides, N1 and the later response. So I guess this is what we start with again next time. So these are sensory event-related potentials. Visual, somatosensory, auditory. I haven't done auditory myself, but I think I always want to, because I want to do mismatch negativity with auditory and not with somatosensory, because somatosensory is a little bit annoying. Yeah, so let's stop here and then continue next week. As for the study questions, I actually didn't say that. I looked over them, so all of them, so the 5 ones that are referred to the random fields, I think you should be able to answer them based on reading the text. And I will also, maybe, let's say the first 4 of the calculus study questions, we should also discuss selectively next time. Yeah, so look into the calculus. Good. Then see you next week.